Thank you very much. That was great. Okay, so now we're going to have industry perspectives. So it's my pleasure to invite Dan Chen to the podium, Global Head of Cancer Immunotherapy at Genentech, to give us his thoughts.
Thank you very much. It's my pleasure to be here and invited by the organizers to present the Genentech perspective on combination development with PD-1 or PD-L1 inhibitors. As such, I will try to present our perspective utilizing data that we've generated and questions that we've generated internally for combination development, primarily with atezolizumab. However, I think many of these concepts, if not all of these concepts, are applicable to the entire field. This presentation is representative of work that I've done now with Kathy Winson, who is our global regulatory franchise head at Genentech for atezolizumab. I did put together 20 minutes worth of slides, so I'm going to move quickly through a select number of them, and this is one of them. We recognize that in combination development for PD-1, PD-L1 inhibitors, that this is a complicated space, complicated by a number of things that we've already started to talk about and will continue to talk about today. And so for this presentation, I put together three main questions. What are the unique regulatory challenges for PD-1, PD-L1 combination therapies? How can the impact of a second drug be assessed when combined with an existing effective drug? Is there a threshold that that combination needs to meet? And how do the information needs and decision-making differ from strategies for developing novel combinations as opposed to therapeutics that you're putting in combination that you already have a pretty good understanding of? So starting here with the first section, let's talk about exploration of general CIT combination. So what are the unique regulatory challenges associated with PD-1, PD-L1 combination therapies? Well, we recognize a few driving factors in this space. PD-1, PD-L1 inhibitors have shown themselves to be broadly active across human cancer in the clinic. We recognize that the immune-based biology is complex. And finally, there's a massive amount of orthogonal in-pathway data. These things are all important factors as we think about the challenges we face with combination therapy. This slide just represents the complexity of the biology. I think most people here recognize two publications that Ira Melman and I have made, Cancer Immunity Cycle on the left, Cancer Immune Set Point on the right. These two frameworks really define a lot of the complexity that is a core part of cancer immune biology. And we need to recognize that this is based upon work of decades worth of work of investigators in this field. So how do we think about combination therapies? I think that there are a few simplistic ways we can start to think about how to put together combinations and bucket them. There are combinations with standard of care. These are probably some of the most direct, one of the most direct categories in terms of combination immunotherapy. So combine with something that is already active and in clinical use with your immunotherapy agent. The next would be a combination with an established in-class therapeutic. Example would be bevacizumab in frontline renal cell carcinoma or bevacizumab in frontline HCC, because there is ample data for both the pathway and the drug in the disease, though neither are approved in those indications as a single agent. The third bucket would be combination with an established agent, but in an indication where it's not established. So again, using the bevacizumab example, bevacizumab we know is broadly active in many different tumor types. It's labeled in quite a few tumor types. It's not labeled in melanoma. That doesn't mean that bevacizumab wouldn't be active as a single agent or in combination therapy in that disease, but it's not approved there. And then finally, combination with a new molecular entity, an entity obviously that would not be as well understood as any of the above. This slide I'm going to move through quickly. These are additional considerations. We recognize endpoints are important. We recognize that the indication that you go into for combination development is important. And then the final of the consideration slides, the different approaches. You can add to standard of care. That's a very straightforward approach. You could add to standard of care, but also test contribution of parts in your pivotal study. And finally, you can take an approach where you try to replace a standard of care regimen with your combination regimen. These are all different in terms of patient numbers, time, cost, and your ability to fully assess in a statistically significant manner contribution of the individual parts of a combination regimen. So with that, I'd like to now move to an example of combination development for PD-1, PD-L1 inhibitors. And I'm going to use the example of Empower 150. This was a very large global phase 3 study. It was 1,200 patients, 3 arms, and it tested chemotherapy plus bevacizumab versus chemotherapy, bevacizumab, and atezolizumab versus a third arm, which was a knockout of bevacizumab with atezolizumab. So 3 different regimens being tested in a single large pivotal phase 3 study. This regimen, of course, utilized 2 chemotherapeutic agents and 2 different biologics. And at the time that it was designed, it was one of the first frontline non-small cell lung cancer combination cancer immunotherapy phase 3 studies in an all-comer population. It was, in fact, the first to read out as such in this indication. And I put that there because it's important to recognize that when the study was designed, there were a lot of questions with almost no answers. No answers around, does chemotherapy combine effectively with immunotherapy? Does an anti-angiogenic agent combine effectively with immunotherapy? But when we think about it, this study very much was based on strong scientific hypotheses. And in fact, the base hypotheses here, chemo combo with immunotherapy and anti-angiogenic combination with immunotherapy, were 2 of the first scientific hypotheses that we went into the clinic with when we initially went into the clinic with atezolizumab. So this was a core foundational scientific hypothesis, and it was based on a large body of preclinical work and biologic studies that had been done leading up to that time. And for those that are familiar with the field of anti-angiogenics, one recognizes that even though we spent probably a decade and a half of development of bevacizumab as an anti-angiogenic, there were multiple labs that were looking at it as a biologic modifier of immunity. And that's reflected here. If we look at the biologic hypotheses around VEGF inhibition as it relates to cancer immunity, we recognize that VEGF is a central mediator of not just angiogenesis, but tissue and wound repair. And a major part of the tissue and wound repair program does relate to its impact on immunity. And it has 3 particular areas biologically. It can impact priming of an immune response that happens very early in the cancer immunity cycle. It can impact T-cell infiltration into the tumor microenvironment that happens at the endothelial cell border. And finally, in the tumor microenvironment itself, a particularly interesting biologic hypothesis, because this is an area that we've actually seen a lot of clinical data now supporting this piece of the biology, which is the effect of VEGF-R and VEGF on a set of immune cells that are regulatory in nature, in particular regulatory T cells and myeloid-derived suppressor cells. Both of these cell types express VEGF-R, are sensitive to the presence of VEGF, and proliferate and exert additional immune suppressive mechanisms in the presence of high VEGF. So this is the design of the Empower 150 trial. As you see, it's a 3-arm trial, 1,200 patients. And in addition to what you see on this slide, I will note that liver metastases were one of the stratification factors of the trial because it was felt to be important to answer a particular question as it related to the presence of liver mets for these therapeutic regimens. In addition to that, it had a perhaps complicated, I would say in-depth statistical design. This allowed us to control for statistical rigor here, maintain alpha control, despite the fact that we had a multitude of scientific questions that we needed to answer with this 1,200 patient trial. It included questions like, is there an important impact on regimen efficacy in patients who had pre-existing immunity or not? Or whether lung patients that had EGFR mutations or ALK rearrangements would benefit from this type of an immunotherapy regimen. It also allowed us to test both progression-free survival and overall survival as part of the statistical analysis, including the comparison between the add-on strategy, adding on atezolizumab to chemotherapy plus bevacizumab, or the replacement strategy, knocking out bevacizumab in the control arm regimen and replacing it with atezolizumab. So that was a lot of questions. This is the top-line data that many of you are aware of. This is updated data that was presented by Dr. Szynski at ASCO and is now published in the New England Journal. You can see that this trial hit on its primary endpoint, both on progression-free survival and on overall survival. And that included in both patients that were EGFR wild type or ALK non-rearranged, as well as inclusive of those two patient populations. Now, these are important findings because there was a control arm that just included chemotherapy plus atezolizumab. And as you see here on overall survival in the ITT wild type, there was certainly evidence of activity for chemotherapy, in this case, carboplatin and paclitaxel in combination with atezolizumab in terms of survival, but not nearly as potent as what was seen with the four-drug regimen. This can be further assessed on an endpoint like response rate. You'll note here that the four-drug regimen of chemotherapy, bevacizumab, and atezolizumab has generated probably the highest response rate that has been seen in frontline non-small cell lung cancer, and that's inclusive of not only CR/PRs, but also CRs. Those CR rates in this disease still remain relatively low. But one of the most intriguing findings to come out of this trial are the subset efficacy data as you compare the three different arms. And what you see here on the left is the overall survival hazard ratio of 0.54 for the four-drug regimen versus just the control arm, a three-drug regimen with chemotherapy and Avastin. This is an important finding, given the fact that we just have not seen this kind of efficacy for checkpoint inhibitors, or even yet reported for checkpoint inhibitors with chemotherapy. We recognize that the liver is a privileged space for cancer immunotherapy, and while you can see responses, you can see benefit in some patients with liver mets, I think as has been noted previously here, we have across the field seen less activity in patients with liver mets with cancer immunotherapy. And again, on the right, when you omit the bevacizumab in the treatment arm, this is now atezo plus chemotherapy versus bevacizumab plus chemotherapy. You just don't see the same kind of survival benefit in these patients with mutant EGFR or ALK mutation. Similarly, when you look at liver metastases, you see a very similar finding. Again, the four-drug regimen showing an overall survival hazard ratio of 0.54, whereas the overall survival for the knockout comparison, 0.85. And always important as you make observations, no matter how strong your statistical analysis plan is, you always want to see whether there is existing data or additional data that can be generated that can also support the underlying observation and hypothesis. And in fact, in this case, for both EGFR mutation and for liver metastases, data in this field has already been generated. At least similar data. If you go back to the original chemotherapy plus or minus bevacizumab trial in frontline non-small cell lung cancer, you'll note this forest plot where patients with liver metastases, when you gave them the addition of a bevacizumab, you saw a better survival hazard ratio. Similarly, when you look at data for EGFR mutants and the addition of bevacizumab, but in this case, the addition of bevacizumab to erlotinib, a drug that targets the EGFR mutation, Again, you saw an improvement in clinical efficacy. So really, the data was already in the field. It just wasn't in the field yet for cancer immunotherapy. And all of this data together can be interpreted as data that supports that there are underlying immunologic effects of VEGF, and that VEGF inhibition can act to further enhance immune-based efficacy in a regimen as tested in the Empower 150 regimen. And this is a figure that comes from a publication that is in preparation, but just noting the biologic underpinnings of these findings. VEGF is downstream, essentially, of all three of these subsets of patients where they display high VEGF states. This is a safety slide, just to note that while there was strong benefits seen efficacy-wise, the safety was very similar to each of the different components individually. And then moving into just my final section here, challenges for cancer immunotherapy combination development in the future. So how do we look at all of this, all of these considerations, and put it in within the framework of combination development to come? Well, here's one example that is, I think, very pertinent. Here, this is data that was shown at ASCO by Stein et al., frontline hepatocellular carcinoma, the combination of bevacizumab and atezolizumab. You see a response rate in the first 20 or so patients of upwards of 60%. Each individual agent here, bevacizumab or atezolizumab in frontline HCC, is expected to have a response rate in the range of 10 to 15%. So this is well above what one would expect in HCC for either of these single agents alone. As we recognize, the combination of a VEGF inhibitor and a PD-L1 inhibitor has now already been shown to be positive in not just one phase 3 trial in lung cancer with Empower 150, but a second trial that met its primary endpoint in frontline renal cell carcinoma with Emotion 151. And so as we look at this combination, the question will arise, what is the right amount of evidence to show contribution of parts as opposed to the combination efficacy that's seen? Now, a slightly different example would be a new agent. And in this case, I show you data that was presented last year at ASCO for a T-cell engager. This is a CEA CD3 bispecific antibody. It has single-agent activity in MSS colorectal cancer, though one would argue that the single-agent response rate is relatively low. When added to atezolizumab, which biologically you would expect to synergize with a PD-L1 inhibitor because CD3 engagers generally drive up PD-L1 expression when they're able to engage the target cell. You see further increased responses, and you see further enhancement of durability of response. However, we recognize that, again, these are new agents. Atezolizumab is not improved in MSS colorectal cancer. There's very little evidence for strong activity of atezolizumab as a single agent in this indication, and CD3 bispecific is a completely new agent. And so one would imagine that you would look at these two scenarios differently. However, I think for all of us as a community, we need to ask ourselves, what is the right balance in terms of generation of data for contribution of parts? And I know that this is something that the agency takes very seriously and looks at on an individual case basis. One way to approach this would be something that was alluded to earlier today, and that is utilizing platforms that really start to combine two different types of study designs that we've used in oncology. In the past, we've often gone from phase 1 to phase 2 to phase 3 in a stepwise fashion. But we recognize, I think, all today in 2018, that we are sitting on a class of agents that can have a dramatic effect on patients' lives. And in the best case scenarios, these are life-transforming or life-altering effects for these patients. And so I think it behooves us all to be able to rapidly prioritize and accelerate drugs that are transformational. One approach to doing this really is to combine phase 1 and phase 2. and rapidly be able to test multiple hypotheses and allow you to deprioritize combination therapies that are not showing a high level of activity and prioritize regimens that are showing very strong evidence of priority. And this is a different way to think about drug development. I heard one comment earlier today that, we don't want to throw away active regimens, and I absolutely agree with that sentiment. However, you might want to prioritize regimens that are highly active over ones that may be less active. Not throw away the less active agents, but really be very clear about your prioritization. And one of the interesting things about this type of a platform that allows you to combine phase 1 and phase 2 to randomize in a single disease across multiple regimens, is that you really do rapidly generate data for different regimens and can clearly see the regimens that have greater activity. Because you're looking at it against multiple arms. Some arms may represent standard of care. Some arms may represent just single agent to start to generate that single agent activity. But in many ways, the multiple arms themselves start to serve as additional control arms. It gives you greater resolution at seeing the activity of the arms that are more active. And there are ways to further bolster your confidence in data that's generated out of a platform like Morpheus. One way to do so, and I think there is an entire session, or at least a presentation on it tomorrow, is to bolster it with real-world data. If we can start to build in a control arm into these regimens and look at real-world data, that is likely to be able to present quite a bit of confidence in the findings you see out of such early, rapid combination development platforms. So just to finish, there are a multitude of scenarios that cancer immunotherapy combinations fall into as you consider their development. Individual contribution of each component of a regimen can be very important and can be incredibly important in not only teasing out biology, but understanding whether each agent in a regimen is really necessary. But this data can come from multiple sources: historical studies, phase 1, phase 2, phase 3, the same study or a different study, or even a platform-based study. All of these are ways to start to generate a data set so that we can look not just at a single study, but holistically at an entire data set to really understand what the right questions are to be spending our precious patient resources on. And so finally, at the end of this, can real-world data be leveraged? I would hope the answer is yes. Given the level of existing data with PD-1, PD-L1 inhibitors, we didn't really go there. But when you have so many of these agents being developed, many asking similar questions, are we really going to continue to develop these as if they were completely standalone trials and drugs? Or will we really start to draw from the larger field? And I think there was a nice example already made of this in the field of BRAF inhibitors and how one can look at a class as a whole. And finally, what are the additional considerations when developing novel cancer immunotherapy combination studies? That is still very much the next wave to come here. Thank you very much.